Hello and welcome to our channel. Today we discuss the discovery of a molecule that links immune system to the brain. For many years, immune and central nervous system were perceived by researchers as isolated entities. However, growing evidence supports the hypothesis that human brain can be influenced by signals from immune system. The evidence emerged from experiments investigating how manipulation with T lymphocytes can modify brain function, including learning, memory, fear, anxiety, and emotions. The key signaling lymphocytes are CD4 cells, also known as T helpers, a group of lymphocytes that facilitate the development of immune response. But they could be not so helpful, acting as agents in autoimmune disorders or allergies. The action point of T cells in the brain most probably is so-called limbic system, gray matter nuclei located deep inside big hemispheres. And these gray matter regulating memory and emotions. It is well known that patients suffering from autoimmune diseases are more susceptible to mental disorders. And more than that, immunomodulatory therapies for autoimmune conditions might aggravate psychiatric problems. This is, for example, true for interferon beta, that is currently used as an effective treatment for multiple sclerosis. Multiple sclerosis is autoimmune disease, but its use is limited by increased incidence of depression and suicidal thoughts in treated patients. In the study we're discussing today, researchers, in order to make animal models of autoimmune disease, created transgenic mice overexpressing an interesting molecule in their T cells, so-called annexin A. There's a whole family of annexins, and more detailed discussion of these molecules we will get in our future webinar, so stay tuned. T cells overexpressing annexin A show clear pro-inflammatory phenotype. And mice with these T lymphocytes showed an increased inflammatory response in the model of autoimmune inflammation of brain and spinal cord. And this line of experimental animals was suggested as a model of systemic lupus frequently seen in humans. It is interesting that on direct observation of annexin A1 overexpressing mice revealed signs of compulsive behavior, such as increased tendency to dig or plucking of fur from cage mates. This represents a model of human trichotillomania, obsessive pulling of own hairs, and other compulsive disorders. A comparative analysis of gene expression in the brain of such mice showed significant differences in level of expression of 15 genes, and several genes of these 15 were associated with emotional disorders. One of these genes in CD4 T cells is responsible for a small protein of size like majority of cytokines. It was named immunomodulin to reflect the possible function of this protein. This protein could be found either in intracellular vesicles, as you can see here, or it could be secreted in the bloodstream. And probably this new discovered cytokine affects the level of anxiety and induces compulsive behavior in animals. For example, mice, normal or wild mice, injected with this molecule showed significant increase in anxiety-like behavior. This change occurs approximately in 7 days after an injection. It shows that the function realized through through modification of gene expression. Researchers tried to confirm that immunomodulin was indeed responsible for anxiety-like behavior by injecting mice with an antibody against this protein. The antibody should bind the protein and clear it from the blood. And mice that received the anti-mood antibodies showed significant decrease in obsessive-compulsive behavior, as you can see here. And now from mice to men. Blood mononuclear cells, lymphocytes, and macrophages of 20 OCD patients were screened for specific cDNA, DNA coding of this protein. And blood from healthy controls was also tested. And the mononuclear cells from OCD patients show high level of the molecule, approximately 6 times higher than in healthy subjects. Today, we don't know yet the target of this protein in the brain or in other organ. And its actual function is to be investigated in further research. So that's all for today. Thank you very much. Feel free to ask any questions, stay with us, and subscribe to our channel.